The archegonial stalk is longer than the antheridial stalk. The venture close the ventral canal cells and egg is the egg cells also there. So in the neck canal cells, around the 6 neck canal cells are there. The antheridia and archegonia are closed in the perigonial and pericytial leaves are there. On maturity, neck canal cells is disintegrated and to form the mucilaginous mass of the cell. Apical cells of the archegonial neck is separated. Anthrozoids are attached to archegonia, swim through the neck and reach the ventral cells, and anthrozoids fuse with the egg to effecting the fertilization. You know that one. So after fertilization, the zygote then divides number of times periclinal and enlarges and secretes the wall around it. Okay. And it will divide transversely into two cells. So both cells are obligated to interesting walls and lead to the two faces of the apical cells are.
Hello. Hello, sir.
Oh, okay. So some hello. Okay, clear?
Okay, sir. Okay, clear, sir.
Okay, thank you. So then the upper cell maybe is divide and gives rise to the capsule and upper part of theta. So see here the development of the sporophyte of the funaria. This one he is this here. That is the see the funaria. This one is the after fertilization is archegonia undergoes to the epibasal cell, hypobasal cell again to formed as a sporophyte like this. Now okay. Hello. Hello, please
Okay,
clear.
sir. Okay, sir.
Okay,
Okay,
thank you.
okay, sir.
Some disturbance are there. Okay. Mm, yes. And the cells are produced from the lower apical cells to the lower part of the theta and foot. The venture of the archegonium grows to form the protective covering called calyptra. Calyptra is also there. What is the structure of the sporophyte of the funaria? A mature sporophyte has the basal foot and thin elongated seta is also terminated in the appear shaped as a capsule. Pear shaped structure is there, the capsule. Okay. Then The food is the conical and small absorb the nutrition and water from the gametophyte. Food is the embedded in gametophyte. Okay, and the supplies to the sporophyte. The seta is the long twisted and raised the capsule above the gametophyte. It is the middle conducting tissue. Means conducting tissue means between the foot capsule, foot and capsule. Theta is the conducting tissue, sterile tissue, which is to connect the foot and capsule. Okay, and the capsule is the differentiation into a basal salon apophysis, middle theca. Column structure is there in the lower part, that will be apophysis, and middle part is the theca, and the apical cap like structure is there. That one is the operculum. See here in the diagram. This one is the, see here, apophysis, and the operculum and opening part is there on the apical region. Okay, clear? Some members. Hello, please, clear?
Yes, sir, clear.
Okay.
Hello.
So all of you keep the mute.
Okay, sir.
Okay. So, and the theta is long, twisted, and this is the apophysis, and theca is there. The theta is the apical portion is expanded from the apophysis. The cells have chloroplast. The epidermis has some stomata are present. Central conducting strands of the theta is extended into the apophysis. This is the fertile part of the capsule is the theca. Theca is covered with the epidermis beneath the, which is the hypodermis is also present. Below the hypodermis, the chlorenchyma tissue is also is there, which is followed with the air cavities, which is the differentiation to some chambers are there, that is the trabeculase. So columella is the central part of the theca. This one is so sterile tissue. So this is surrounding the columella is the barrel-shaped spore sacs also present. Both sides of the columella, two spore sacs present. Four mother cells present in the spore sac undergoes to the meiosis division to form the haploid spore, which is developed in this spore sac. So each spore has the outer colored wall is the exposerium, and inner one is the endosporium is present. So nucleus and oil and globulus and chloroplast also present in the each spore. In the apical part of the capsule is consisted operculum and peristome is also present. So some operculum and peristomes also present. Operculum is 4 to 5 layers in the thickness. Just beneath the operculum is the peristome is also there. The peristome has two rows of the 16 curved triangular teeth present in the peristomes. So outer teeth are red in color and inner one is the colorless, pale colorless are there. The inner teeth are shorter than and outer teeth. So these peristomes are used in the dispersal of the spores. 16 peristomes are there. Peristome trees. And dehiscence of the capsule. As the capsule is the maturity stage, the cells of columella, operculum, base of the annulus and other walled tissues are capsule lose the water and which will be dispersal of the spores. Okay. This result in the rupture of the annulus and operculum. As soon as the operculum is detached, the hygroscopic peristome teeth, which help in the growing area the operculum and also help the spore discretionation. So it is also hygroscopic nature is there. So which are helps in the spore dispersal. Usually spores undergo period, some resting period is there. When the favorable condition, they are germinate in the within the few days. The outer wall of the spore is rupture and inner one to produce the germ tube. So after the germ, a protonym is developed. A filament-like structure, primary protonym, multicellular primary protonym is developed. The primary protonym is differentiated into after some rhizoids and stems and leaves will be developed as a complete uh, finaria. Okay. The next one is the evolution of sporophytes in bryophytes. So what is the evolution is there in the sporophyte bryophytes? So there are the see here. So eight instruments are there. So the evolution of the sporophyte in bryophyte. This is the though structurally there are the differentiation is there. So here so here the evolution of sporophyte in bryophytes, there are the two theories are there to explain the development of the sporophyte. See here from the Markantia, we see here from the from Markantia means earlier the Rixia is also there. So in the Bryophytes, there are the two markable alternation of the generations are there. Yesterday we discussed that one. Gametophytic phase, another one is the Sporophytic phase. Two stages are there in the life cycle of the Bryophytes. A series of the Sporophytic generation which is in the these Bryophytes. There are the different structures are there, sterile structures are there, fertile structures are there. So according to their complexity, means complex structure of the Sporophyte, here they are arranged in the a series from the simple to the complexity or complex structure. This one is the start from the simple Sporophyte is the Rixia through the Markantia, Pelia, and Spiras, and finally complex structure of the Sporophyte in Funaria. This is the evolution of the Sporophyte. Means simple structure of the Rixia is present, and complex structure means the structure of the Sporophyte is the complexity is there. Means other tissues, sterile tissues also present, including the fertile tissue along with the fertile tissue. So that's why there are the theory of the sterilization theory. They are explained, the two theories are there. So these sterile theories explained by the Bower. So this one is also called as the progressive sterilization of the potential fertile instead of the forming the spores of uh, propagative potentially in the fertile cells. So that's why this theory is mainly explained in the Bryophytes in the basis of progressive elaboration and complexity of the Sporophyte. There are other some members are there, Campbell and Smith. There are the followed the Erixia, Markantia, Pelia, Anthoceras, and finally Funaria. So what is the Sporophyte of the Rixia? We can see here that after zygote means after fertilization, they are increased in size, undergoes to the single division forming the two-celled embryo is there. This embryo is repeated division to form the 20 to 30 undifferentiated cells are there. That is the outer one is the amphithecium and inner one is the endothecium. The amphithecium to form the single layer capsule wall, but the endothecium became the archisporium means spore mother cell. So spore mother cells to produce the spores. So in Rixia, we cannot see the foot and seta. Only the capsule is there, means sporogenous tissue is present. Circular sporogenous tissue is there. That is the simple structure in the Rixia. So another Markantia. So in that one, here there are the two-celled embryo divide right angle to the to form the four-celled embryo. So these out of the four cells, two lower one and they are called the hypobasal, and upper one they are called the epibasal cell. Okay. So this one is the capsule is portion is differentiated to amphithecium and endothecium. The single layer capsule wall is formed from the amphithecium, and the entire mass of the endothecium act as a sporogenous tissue. So from these of the archisporias remaining some sterile tissues, elaters also there. So this one is the spindle-like structures are there. This one is hygroscopic nature, helps in the spore dispersal. You know that one. The Markantia sporophyte, the broad foot and well-developed seta is also there. Single layer sterile jacket layer is capsule is present. So sterile apical cap is the apex and spirally thickening elongated elaters also present. The next one is the pelia. So what is the Pelia structure means? Hypobasal cells does not take part of the development of the sporophyte. Uh, the epibasal cells divide and differentiate into outer amphitheecium and inner one is the endothecium. The amphitheecium forms the capsule wall and the endothecium act as the archisporium. The amphitheecium forms the capsule wall and endothecium act as the archisporium. You know that one. The upper part of the epibasal cells to form the capsule and lower, that is the sterile elaters are there. So the elaters are long, slender with the bulbal double spiral thickenings also present. Double layer thickenings are there. Some of the sterile cells became the compact arranged as elatophore. Elatophores also present. So means here the pilia that means the complexity means sterile tissues are there. The next one is the sporophyte of the Anthocera. So here, two-celled embryo is converted into eight-celled embryo and arranged into two tiers. The lower tier by further division to form the mass foot and the cell is the upper tier also repeated division to form the capsule. The capsule is the young condition is differential into outer amphitheecium and inner
Sir,
endothelium.
disturbance వస్తున్నాయి సార్. సార్,
Hello? Okay.
disturbance వస్తున్నాయి సార్.
Okay. Sorry. Now clear? Hello.
ఓకే ఓకే సార్.
Okay.
ఓకే.
So the amphitheecium, the archisporium form the spore mother cells and some solo elaters also there. The archisporium in the Anthoceras thus extremely is reduced. It forms the narrow part of the capsule and covering the outer layer. The archisporium is derived from the amphitheecium. The jacket layer, it consists of chloroplast and stomata. So this unable to sporophyte lead the independent existing is there. But Markantia, Pelia, and Pixia, they're completely depending on the Gametophyte. So this one is the Anthoceras means they are completely. So this type of the sporophyte shows remarkable reduction in the sporogenous tissue due to the more sterilization. Sterile tissue is there. So centrally columnized, yesterday we discussed centrally columnized there. And hypodermis is also there, means sterile tissue is more. Next one is the funaria structure. So major part of the sporophyte remains sterile and to form the food seta. Major part is sterile tissue. The capsule of the sporophyte is developed to form the epibasal cell, foot and seta from the hypobasal cell. The epibasal cells divide periclinally, forming outer amphitheecium and inner endothecium. The amphitheecium becomes differentiated into epidermis, that is the photosynthetic capsule wall and outer spore sacs also. The amphitheecium at the apex of the capsule forms the multi-layer operculum, while the innermost portion of the peristomes. The endothelium is sterile and thin outermost layer form the archisporium. The capsule is the pear-shaped structure is there. The basal sterile is known as the apophysis. Apophysis totally sterile tissue which is involved in the photosynthesis. The capsule is the pear-shaped structure with the basal sterile. That one is the apophysis are there. So outermost layer is the epidermis. That layer which is the parenchyma tissues. That parenchyma tissues is divided in some air chambers also there. Between the air chambers, a flamen-like structures are there. That one we call as the trabeculae. So surrounding the lid of the capsule, a ring-like circulated structure epidermis known as the annulus is present, which is helps in the spore dispersal. So the progressive sterilization there is potentially sporogenous tissues increased here. The gives rise to explain the Bower theory sterilization of the evolution of sporophytes in bryophytes. So another theory is also the reduction theory. So this another school reduction theory means Church, Campbell, and Evans. They hold that the evolution and the down directions are the series of the retrogressive evolution. This is the evidence of this type. The reduction is accompanied by the simplification of the structure of the sporophyte. Means here complex to simple structure. So that's the reduction div. On the basis of this simple sporophyte, Rixia is considered as the highly evolved through the reduction as a result of the progressive simplification. So some steps also there in the reduction descending evaluation. The leaves erect support sporophyte were the lost due to the intense isolation of desiccations. The erect leafy sporophyte became attached to the gametophyte. The airspace, the photosynthetic system disappeared due to the more dependence on the gametophyte. Stomata first became functionless and later it is disappeared in the Markanch and younger Malians. So simplification of the dehiscence operates are present. So reduction the photosynthetic tissue and some these changes are the progressive increase in the fertility of the sporogenous tissue. So this evidence to from the comparative morphology experimental genetic support in the view of the Rixia sporophyte is advanced based on the structure. Okay. So evolution of sporophyte is the compulsory question in this syllabus. Okay. So next one is we discuss the pteridophyta.
Sir,
So
can
what
you
is
please explain what is amphithecium and endothecium?
so amphithecium? That is the means amphithecium is the outer layer. After embryo, number of divisions are takes place. They can divide into two parts are there. After zygote undergoes to the divisions. So to form the two layers of the cells are there. Amphithecium is the outer layer and endothecium is the inner one. Okay, amphithecium and again they can divide as the foot and seta. Endothecium which is developed as the archisporium means nothing but sporogenous tissue. Clear? Okay.
Thank you.
Okay. So the next topic we can discuss the pteridophytes. So what is pteridophytes means here general characters and classification of the pteridophyte. Means these pteridophytes are the first vascular plant. First vascular plants we call as the pteridophytes. Here, so pteridophytes, there are the two Greek words are there. Peris means fern-like structures. Phyta means plants. So fern-like structures of plants include in the pteridophyta. So which is include around the 10,500 species and 400 genera are there. There are the vascular cryptogram. They call as a vascular cryptogram. Means first of all, vascular structure is there in the pteridophyta. They are possessed means conducting tissue, xylem and phloem is there. So there is no flowering. So that's why they call as a cryptogram. So this division of the pteridophyta placed between the division of the bryophyta and the spermatophyta. There are more advanced than the bryophyte and primitive than the spermatophytes. So the pteridophytes are widely distributed in temperate zone as well as the tropical regions also there. So these pteridophytes, some of the living genera are present. For example, Rhinia. There are the in the identified in lower and middle Devonian period. So these pteridophytes are dominantly present in Carboniferous period around the 35 crores of the years back. Lower Devonian to middle Devonian period. That is the dominant period. Carboniferous period is the dominant period. So the pteridophytes there include the club mosses are there, quillworts and arch tails. So club mosses means Lycopodium selaginella and quillworts Isaitis. There are like a quillworts are arch tail like structures are there, equisetum and ferns also there means Marsilia. So based on their structures, different structures are there. So what is the characters of the pteridophyta? Suppose there are two distinct generations are there, that is the sporophyte and gametophyte. So both the gametophyte and sporophyte are the free living individual plants are there. There are independent plants. There are the independent. They are alternate each other. So that's why the alternation of generation we calling that. So here sporophyte of the pteridophyte, the plant body varies in size and structure of the among the pteridophyte. This one is the differentiated into roots, stems, and leaves are there. Okay. So most of the living sporophytes, these pteridophytes, the herbaceous structure is there. Herbaceous like structures are there. Mm. So the stem is mostly herbaceous and aerial structure, but the fern in the either horizontal or oblique and rhizomatous structure. Rhizomes also present. Okay. The vascular system of the pteridophyte consists of xylem and phloem. The xylem possesses the tracheids and vessels are absent. Only tracheids are present. The phloem consists of sheep cells and phloem parenchyma companion. These cells are absent. So this one is the stele is protostele. The means protosteles means there is no pith in the center of the uh, stem. Means that one we call as a protostele. Only the xylem is surrounded to the phloem. That's why there is no pith is there. It is not present. That's why we call it as a protostate. So the most of the pterophytes homosporous condition is there. There are produced only one type of sporangium. Uh, that's one we call as a homosporous. Lycopodium is the homosporous condition. And sometimes some type of the ertospores also there. Heterosporous. Megasporangium and microsporangiums are there. Megasporangium is the female reproductive and microsporangium is the male reproductive. Like that. They are active. So that condition is the heterosporous. For example, Selaginella marsilia. This is the heterosporous conditions. Gametophytic condition means haploid spores are the pioneer of the gametophytic generation. The gametophyte is the pterophyte is the simple dorsiventral thallus and undifferentiated. This one is known as prothallus. So some mycorrhizal association is also present in the gametophyte. So the gametophyte, they bear the male and female reproductive organ, that is the antheridia and archegonia. Like the structures are there. So after the fertilization, there are the pteridophyte, similar fertilization is there between the pteridophyte and the bryophyte. After fertilized egg, they develop into embryo and the prothallus tissues are there for the nutrition and development of the embryo. So diploid sporophyte, the embryo is shows the food, root, and leaves, stem in shell within the there in the after development. So what is the life cycle of the pteridophytes? Means heteromorphic alternation of generation. We calling that heteromorphic alternation of generation. Means morphologically different phases are there. That is the sporophyte and gametophyte. That's why heteromorphic we calling that one. So the sporophyte result to form fusion of gametes and to produce the sporophyte. This one. So what is the apogamy? Apogamy means there are a Greek word is there. Apo means without, gamy means gametes. The formation of the sporophyte directly from the gametophyte without gametic fusion. That one we call as apogamy. Without gamete fusion. So directly gametophyte to form as a sporophyte. This is a special character in this pteridophyte. Any vegetative cells may be involved in this process and to form this sporophyte. That one is the haploid condition. In the example, lycopodium. Apospore. Apo means this one is also derived from the two Greek words are there. Apo means without, spore means spore. Means without spore formation. So this one is developed through the gametophyte from the sporophyte without spore formation. So this gametophyte, this sporophyte is haploid condition is there. It may be developed from the vegetative cell of the sporophyte, thus reduced in the gametophyte, which is a diploid condition is there. So this is the apogamy and apospore. Apogamy means without gametes. The gametophyte to form as a sporophyte. Apospore means without spores, sporophyte to form the gametophyte. This is the structure of the apospore and apogamy. So next one is the classification of Smith is classified. The different divisions are there. Finally, Smith is Sporne classified. Legend of different classes are there. So finally, Sporne in 1968 is divide the pteridophyte into 5 classes are there. That is the Silopsida and Silotopsida and Silopsida, Lycopsida, Spinopsida and Pteropsida. These 5 classes are there. What is that one? Based on their structures. What is that one? Silotopsida, Silotopsida, Lycopsida, Spinopsida and Pteropsida. This one is there. So from that one is Silopsida. There are the Silotem is there. And another division is the Lycopsida. Lycophyte or Lycopsida. Here, this one is the Lycopsida, Ligulopsida, and another one is the Iligulopsida. 2 classes are there. Another division is the Spinopsida, Spinophyta or Spinopsida. This is Equisitum. Okay.
సార్ వీడియో వస్తలేదు.
Hello.
వీడియో వస్తలేదు.
Okay. Okay. So this one is the some yesterday before that one we discussed that one. Sporophylls to bear the sporangiums are there inside the sporum. This one. Okay. So another class is the Pteropsida. Another division is the Pteropsida. This Pteropsida, then Cladophytales, that is the Cladophyllon, and another one is the Ophioglassales. The Ophioglassum we discussed that one, and Osmunda, Marsilia, and Salvinia. These are there. Okay. In the next class, we discuss the some type studies of the pteridophytes. Okay, thank you.
Thank you, sir.